Welcome to this lecture on cell and molecular biology. Today, we are diving into one of the most fascinating topics in biology, gene regulation. We will explore how activators, repressors, enhancers, and silencers work together to control which genes are turned on or off. Understanding this is the key to understanding how a single cell can develop into a complex organism. So, let us get started. To understand how gene regulation works, imagine you are driving a highly advanced sports car. The engine of this car represents RNA polymerase, the basic machinery that reads your DNA. But an engine running at a slow idle is not very useful on a highway. You need a gas pedal to speed up, which in our DNA are special sequences called enhancers. You also need a brake pedal to slow down, represented by sequences called silencers. However, pedals cannot press themselves. They need a driver's foot. In your cells, specialized proteins called activators press the gas and repressors press the brakes. Now that we have our car analogy, let us look at the formal biological definitions. Enhancers and silencers are specific non-coding regions of your DNA. Interestingly, they are often located thousands of base pairs away from the actual gene they regulate. Because these regulatory sequences are located on the exact same DNA molecule as the gene itself, biologists refer to them as cis-acting elements. The prefix cis means on the same side. These regions act as the physical control panels embedded directly within the DNA strand, waiting for the right signals to be activated. While the control panels are built into the DNA, the feet that press them are separate molecules. Activators and repressors are specialized proteins known as transcription factors. Unlike the DNA sequences we just discussed, these proteins are synthesized elsewhere in the cell and must travel to the DNA to do their job. For this reason, they are called trans-acting factors, with trans meaning across. When an activator protein binds to an enhancer, it boosts transcription. Conversely, when a repressor protein binds to a silencer, it blocks the machinery or tightly coils the DNA, effectively shutting down gene expression. Since enhancers can be thousands of base pairs away, how do they actually communicate with the gene? This diagram illustrates the elegant solution: DNA looping. Look at the blue enhancer region on the left. When the activator protein binds to it, the flexible DNA strand physically loops around. This looping brings the distant activator into direct contact with the promoter region, shown in green. Here, it helps recruit RNA polymerase and other transcription factors, forming a large active complex. This physical connection is what allows the gas pedal to rev up the engine of the target gene. Understanding this looping mechanism brings us to a crucial question. Why is this regulatory system so important? It actually explains the fundamental mystery of multicellular life, which is cell differentiation. Every single cell in your body, whether it is a neuron in your brain or a muscle cell in your heart, contains the exact same DNA manual. The only reason they look and act differently is because different sets of activators and repressors are present in each cell type. They turn on different combinations of genes. If this delicate balance breaks down, for example, if a mutation causes an activator to constantly bind to a growth gene, the cell might divide uncontrollably, leading to severe diseases like cancer. To truly understand how activators turn on genes, we must look at the mathematics of protein DNA binding. This brings us to the concept of fractional occupancy, which is the probability that an enhancer is actually bound by an activator. The formula is Y equals the concentration of A divided by the sum of KD and the concentration of A. Here, Y is the fractional occupancy, a number between 0 and 1. The symbol A in brackets is the concentration of the activator protein inside the nucleus. KD is the dissociation constant, which measures how tightly the activator grips the DNA. A lower KD means a stronger grip. If the activator concentration increases, Y increases, turning on more genes. Now that we have our formula, let us apply it to a specific example. Imagine a gene responsible for muscle contraction. We are given that the activator protein has a dissociation constant, KD, of 20 nanomolar. The current concentration of the activator, A, is 60 nanomolar. We need to find what fraction of the enhancers are currently bound. We start by substituting our given values into the binding equation. This gives us Y equals 60 divided by the sum of 20 and 60. Adding the denominator gives us 60 over 80. Dividing these numbers yields a final fractional occupancy of 0.75. Physically, this means that exactly 75% of the enhancer sequences in the cell are currently bound by the activator protein. So we have calculated that 75% of the enhancers are occupied. This brings us to the next logical step. What does this occupancy actually do to the gene? Once the activator successfully binds to the enhancer, it immediately begins to increase the rate of messenger RNA production. We can model the total transcription rate of the gene based directly on this fractional occupancy. This allows us to connect the physical binding of the protein directly to the biological output of the gene. In most simple models, this relationship is represented as a linear increase, starting from a slow, baseline default speed. This linear relationship is captured perfectly by the transcription rate equation. The formula is R equals R basal plus the difference between R max and R basal multiplied by Y. Let us break down these symbols. R is the total rate of transcription, meaning the number of messenger RNA molecules produced per minute. R basal is the slow default speed at which the gene is read when no activators are present. R max represents the absolute speed limit, the maximum rate when every single enhancer is bound. Finally, Y is the fractional occupancy. In plain English, the actual rate is just the baseline rate, plus a bonus amount of transcription that depends directly on how many enhancers are activated. Let us see this formula in action by continuing our muscle gene example. We are given a basal transcription rate of 5 messenger RNA molecules per minute and a maximum possible rate of 45. We will use the fractional occupancy of 0.75 that we calculated earlier. We want to find the actual transcription rate right now. First, we substitute our values into the equation. We find the difference between the maximum and basal rates, which is 40. This 40 represents the maximum possible bonus transcription. We then multiply this 40 by our occupancy of 0.75, which gives us 30. Finally, adding this bonus of 30 to our basal rate of 5 gives us a final answer of 35 messenger RNA molecules per minute. Before we conclude, let us take a moment to interpret what that number 35 actually means for the cell. The presence of the activator protein successfully boosted the gene's output tremendously. The gene went from a sluggish baseline of just 5 molecules per minute to a highly productive 35 molecules per minute. This massive increase ensures that the muscle cell gets the large amount of proteins it needs to contract and function efficiently. It is also worth noting that if a repressor were to bind to a silencer instead, it would mathematically drive this rate down, pushing it below the basal level and toward zero, effectively silencing the gene. We have covered a lot of ground today, so let us review the key takeaways. First, we learned that enhancers and silencers act as the regulatory control panels embedded directly within the DNA strand. Second, activators and repressors are the trans-acting protein messengers that bind to these panels to either crank up or shut down gene expression. Third, we saw how DNA looping physically connects these distant elements. Finally, we used mathematical models like fractional occupancy to predict exactly how a gene responds to cellular signals. By mastering these interactions, you now understand the fundamental mechanism that allows a single fertilized egg to develop into a complex human being. Great job today.